Started. Morena, no mai, haere mai, ki te hui Aotearoa, New Zealand Food Safety Science and Research Centre. Ko Vivian Hunt tēnei. Kia koutou katoa. So my name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science and Research Centre, welcome to the second of our Spotlight on Food Safety Science seminars for 2021. Today we're going to talk about the seminar is reporting on some work initiated by the Centre to learn more about controlling Campylobacter in poultry, both on farm and during processing. This work is being done with PANZ, which is the Poultry Association of New Zealand, which is represented by most of the New Zealand poultry sector. And it draws on multiple scientists, including Professor Nigel French from Massey University and Patrick Biggs, and a team from ESR, which is Joe and Rob Lake, and also contributors from MPI. I'll let Joanne give the credits at the end, but Joanne is going to lead this seminar talking about this study. Joanne Kingsbury is a food microbiology senior scientist at the Risk Assessment and Social Systems Group at ESR. She earned her PhD from the University of Canterbury in cellular and molecular biology in 2000 and worked for 16 years at Duke University Medical Center in Durham in North Carolina, where she was studying fungal molecular biology and models of disease. Coming back to New Zealand 5 years ago, she joined ESR, and her work today focuses more on understanding and detecting, managing the risk of disease causing microbes such as Salmonella, Campylobacter, and Listeria in the food and food production environments. Jo has contributed to lots of the work that the Centre runs, and she's also a collaborator on the MBE funded project that looking at the impacts and mitigation of microplastics in the environment. We have on the call today too Roy Biggs, who has been a major contributor to this project. Roy is formerly the Quality Assurance Manager for Tegel Foods, and his expertise is in Campylobacter and Salmonella control and Listeria control. Roy is a fellow of the New Zealand Institute of Food Science and Technology, and in 2017 he received the most prestigious award, which is presented by this institute, the J.C. Andrews Award. He's been a member of the IAFP since 2003, and he chaired the New Zealand affiliate for two years. So I think we're going to start with Roy contributing, and then Jo directing action via the slide. So Roy, I'll hand over to you, and just remember to keep your phones on mute while they're talking, and any questions you have, do send via chat, and we'll allow time for that at the end. Thanks very much, everyone.
Thank you, Vivian. Yeah, so next slide, please, Jo.
Sorry, I'm trying, Roy, it's not progressing. Oh dear. Okay, here we go.
Okay, thank you. So you can see in the early 2000s, New Zealand had one of the highest rates of Campy in the world, reaching a peak in 2006. Following the implication of poultry is the most important contributor. The industry made some interventions, mostly at processing level, which resulted fairly quickly in a 50% drop in cases. However, despite the work the industry did, Campylobacter transmission, or 75% of Campylobacter transmission, remains attributable to poultry. Next slide, please, Joe. This is the instance of foodborne Campylobacterosis. You can see it's going down slowly, especially in 2019. And that's despite the increase in poultry consumption, which is the bottom graph there. It's gone from less than 30 kilos up to about 44 kilos over that period. And despite the work the industry's done, sorry, Joe, the SACNZ study still estimated that 84% of human cases could be attributed to poultry. That wasn't the best day of my life when I heard that. Next slide, please, Joe. This is the National Microbiological Database. And you can see on the left, you've got year. To the right of that, you've got the mean log count. mean log counts come down fairly dramatically now. Even if we had no Campylobacter in poultry, the mean log count would still be at 2, because that's the value given to a not detected sample. So you see we haven't got much room for manoeuvre on this system, which is why both the industry and MPI are working hard on developing a new system. The standard deviation has come down dramatically as well, ST, so you can see there's much less variation in the birds than there used to be. The 80th percentile used to be on there, but it's been below the limit of detection for many years now, so there's no point. You can see we've had a 2-log reduction in the 95th and 98th percentile values, so we've got a very significant decrease in the numbers of the bird. Human rates are much more volatile, so no more comment on that. Can you, next slide please, Joe? So the on-farm control of Campylobacter in poultry, it's a longitudinal study through the life of a broiler flock, looking at the various vectors and looking for opportunities for control. Salmonella in poultry, now most of you know me, I think Bigsy's finally lost it. You see him talk about Campylobacter and he's ranting on about Salmonella. But there is a method in this, you know. In 1996, the report from ESI indicated 17% positive for Salmonella. In the USA, the baseline study was going on at the same time, showed 20% positives. At the end of 97, the largest company discovered Salmonella in its grandparent flocks. That was vertically transmitted Salmonella, so of course it went through all of the broiler flocks, unfortunately. In 1998, the CEO of that company demanded action from the management team. Also sent us across the world, a lady called Sharon Wagner and me, and we were looking at Salmonella control around the world. We found a bird rinse method in the USDA ARS in Georgia. And Sharon and I recommended it was adopted because then we had a comparison, as they published the data, between what we were doing and what they were doing. Next slide, please, Joe. Next slide, please. Thank you. This is the Salmonella incidence on carcasses from 2001. It's NMD data. A couple of years before that, it was Teagle data, I may as well be honest. Now, you can see there's been a significant reduction there. And I don't know whether you can read the figures down the left-hand side, but for the last 6 years, it's been less than 0.1% of carcasses have had Salmonella on. In 2019, it was 0. And last year it was back up to 0.05. 2,100 samples per year go into that data, so it's quite significant. I think we should stop here because this is a much better story than Campylobacter. But I thought we've got to go on. So our question was, you know, would the type of work, because I'm dissatisfied here, with the type of work that we'd done on Salmonella, which was basically controlling hatcheries, breeder farms, feed mills, and a little bit on broiler farms. Would that type of work do the job for Campylobacter? Well, it didn't, unfortunately. We've been working on Campylobacter since 2004, when the industry did a survey, and we found 74% of farms. So it wasn't much. In terms, that was about the average for the country data around the world. So we didn't worry too much about it. We thought, all right, we're in line with everybody else. So whatever's happening in New Zealand has got nothing to do with us. But but we developed an enumeration method to do some trials. It might come home at some stage. And then at a famous meeting that I can remember vividly, the industry were presented with the MLST data from Massey team, which was the major source of the high rate of human illness. And Andrew Mackenzie made it very clear either we did something about it or he would. And we agreed fairly quickly on a joint strategy between the industry and MPI. We tested many interventions. ASC, the sulfide sodium chloride, gave an extra 2 log reduction in trials, and 1.6 was installed. That was the best one we found. Okay, Joe, next please. Between 2000 and 2009, we really worked hard looking for controls in agriculture. We couldn't find anything that was working effectively. So it's very different to salmonella. So we concentrated on processing plants with the success that you've seen on the N&D data. And many investigations since, flies, crate washing, breeding efficacy, human factors. Also, we had been following listeria tracking in factories using PFG, E and then WGS. Great success to find out where it was coming from and how we could control it. So we thought, well, can this be used in Campylobacter? So we started developing the current trial. Next slide, please, Joe. So this is Kaipi Farm in Taranaki. It's got a bank on the left and it's got a river about 60 meters away on the right. And in the distance, you can see Mount Taranaki. There's 9 sheds on this farm, each of which contains around about 40,000 birds. So there's a lot of birds in there. Next slide, please, Joe. This is the type of shed we used. It's got a fence down the side. It's 133 meters long and 60 meters wide, with concrete pads on each end. The extract fans are on the end, so the air comes in on the sides of the shed and is pulled out at the ends of the shed. We did many different samples in this farm, and you can see that the number in there, but I know Joe's going to talk about that a little bit later, so I won't spend too much time on that. Next slide, please, Joe. And this is yours.
Okay, great. Thanks, Roy, and thanks, Vivian, as well. So this study was conducted in 2 different phases. The first phase was led by us at ESR, and that involved a literature review by Nikki King at ESR, the development of a Campylobacter specific metabarcoding method by the team at Massey led by Patrick Biggs, and also a limit of detection laboratory study. And the reason for this laboratory study was because a lot of the different types of samples that we proposed to test on the on-farm survey are not typically tested by Teagle New Plymouth, and there are also not necessarily standardized methods available for them. So all of the different projects from phase 1 then informed on the implementation of the phase 2 longitudinal survey on the farm, and this was led by a team at Massey. So first, a little bit about Nikki King's findings for a literature review. She looked at the current literature to see what is known about important reservoirs, pathways, and risk factors for Campylobacter contamination in flocks. And she found that worker movement into sheds was the most important vector for Campylobacter ingress into the sheds. And she found increased risk when neighbouring broiler farms were present, but less so if there are other types of livestock, such as cattle or sheep. And she found some evidence for flock cage, wildlife pests, cleaning, biosecurity and seasonality, but limited evidence for vertical transmission, so in other words from the breeder flocks, or feed, air or drinking water, although there's potential for contaminated standing water around the farm to be relevant. And she also reviewed sampling methodologies that we proposed to use on the on-farm study. Now, a little bit, I'm going to talk about the rationale behind the development of the Campylobacter specific meta-barcoding approach that Patrick undertook, but I'm not going to talk about the actual implementation of this. I'm just going to give an example of this because it's still being reported on, and ideally Patrick will talk to you about this at a later stage. So conventional culture methods can miss Campylobacter in samples, particularly when the Campy is in mixed populations in a single sample, or else when there are low numbers or slower growing genotypes that could be outcompeted during enrichment, or else if the Campy is entered into viable but non-culturable or is now died. So the meta-barcoding approach has the potential to overcome these limitations and could ideally provide a more comprehensive picture of the Campylobacter genotypes present, and thereby increasing the chances for making potential linkages between sources and birds. So an example of what this would look like, you take an environmental sample and extract its DNA, and then you would PCR amplify a region that is specific to Campylobacter but has a lot of heterogeneity across that genomic region. So this was one of the challenges for Patrick to define or decide which area of the genome to amplify. And then you do whole genome next generation sequencing, on the PCR products, and then analyze, compare the sequences that you get out against a reference database, and then assess the Campylobacter allele types and proportions. And in the made-up example that you see below, the blue type from chickens matches what you see from flies, and it's also present in litter and sparrows. So as I said, that's, this study is being reported on, and that's really all I'll say about that. It's just more about the rationale of that part of the project. So for the broiler farm longitudinal survey itself, we followed the life cycle of a single broiler flock, and we took samples all along the way of potential vectors and reservoirs for Campylobacter contamination of flocks, as well as looking for evidence of flock colonization. All of these samples were then sent to the Teagle New Plymouth lab, charged Marie Callender, and Marie did enrichment broths to try and isolate Campylobacter from all of these samples, because a lot of the Campy might be stressed in these samples. She then, for the samples that she isolated Campylobacter from, she sent the isolates to Massey for speciation and whole genome sequencing, and she also sent aliquots, the enrichment broths, to Massey for the meta-barcoding analysis. And then from the whole genome sequencing and meta-barcoding, we ideally would like to link Campylobacter from reservoirs and vectors with what is found in the poultry flock. So a little bit more about the sample types that we tested. We tested the breeder flock as a potential reservoir for contamination of our study flock. And we tested, particularly we tested the flock at the time of the, when the eggs were being laid for our test, our study flock. We tested the previous flock that was in the broiler shed before our study flock, and this was both environmental samples from the shed as well as evidence of samples from the birds themselves at processing. We took quite a lot of samples from the catchers and their equipment. So these are the crews that come in to catch the birds to take them off for processing. We took, for example, cotton glove samples. So these were the gloves that they used in the previous farm for catching the flock that they, previous flocks, as well as swabs from their boots, crates that are coming into the shed to put the birds into in modules and vehicles. And this was done at each cut. There were 3 cuts for the flock. And we also looked for evidence of flock colonization, both from live birds every 5 days doing cloacal swabs, and from cecal samples and carcass rinsates after the birds were in processing. And we took a lot of different sample types from both inside and outside the broiler shed. Inside the shed, we took samples of boot socks from around the shed. We took samples of the actual water itself, the water supply in the shed, as well as swabs from the drinkers, which you can see as a dotted line in the shed diagram below. Swabs, the shed floor and worker clothing, and shed entry. And we also took flies. We caught flies on fly paper inside the shed, crawling insects in a device called an Arends tube, and rodents and traps, as well as samples from outside the shed rabbit and wild bird droppings and soil samples. And we did this every 5 days through the life cycle of the flock. So this, in this example, this is kind of a summary of the sampling and the Campy that we're getting out. So on the, I don't think the point is actually working here, is it? No, so on the left-hand side, you see all the different sample types, and along the bottom, we have the timeline for sampling. When you see a red box, that means Campylobacter was detected from some of the samples of that sample category. When it's green, there was no detection of Campylobacter from that sample category. So the first thing that we see is that the breeder flock was positive for Campylobacter lari, but we didn't isolate this species from any other samples. So the breeder flock was not a reservoir for Campylobacter contamination of our study flock. We tested the previous flock that was in the shed before our study flock, and we found just before they were sent off for processing, the shed environmental samples tested positive, and so did the previous flock. The day-old birds are placed, well, after hatching on day 0. Sorry, I should say that after the previous flock, the shed is then cleaned, and we didn't find any Campy in any of the samples after the cleaning of the shed. So on days 5, 10, 15 and 20, we looked for evidence of flock colonization and took the samples from inside and outside the shed and found no samples testing positive for Campylobacter, except on day 20 when we found some wild bird and rabbit samples testing positive. Days 26 and 28, still no positives from the flock or the shed environment. However, day 29 was our first cut of our study flock. We find that about almost a half of our catcher samples test positive for Campylobacter. And our flock, we find that all cecal samples are still negative for Campylobacter, but 1 out of 15 of the rinsate samples are positive for Campylobacter, which hypothetically could have come from cross-contamination of the rinsates. And we find the same for a control flock that we also monitored that is age-matched with our study flock. By day 35, however, we find that our flock is, both flocks are now Campylobacter positive, and we find a lot of positive samples in our shared environment as well. So in total, we tested 738 samples for Campylobacter and found 26% of these were positive for Campylobacter. The majority of these, the speciation was done by Anne Midwinter in Massey, and she found that the majority of these were C. jejuni. As I mentioned, we only had C. lari from our breeder flocks, and C. coli was mostly coming from catchers as well as some shed samples. So we only sequenced C. jejuni isolates. And the questions we wanted to ask are, what multilocus sequence types are present? Are these the priority types for industry or clinically relevant types? And can we make linkages between isolates from poultry samples with those from vectors or sources? So this is a minimum spanning tree of the whole genome sequencing data for the 199 C. jejuni isolates sequenced, and this was all done by Patrick Biggs at Massey, DNA isolated by Anne Midwinter. And if you're not familiar with this sort of diagram, what you're looking at is each circle is either a single isolate, or if it's a bigger circle, it's multiple isolates. So the bigger the circle, the more isolates are indistinguishable by this method. The branch lengths are the allele differences between the different circles, and these are on a log scale. And the different colors represent the different sequence types identified, and you can see that there were 7 different sequence types among the 199 isolates. The sequence type 6964 predominates. More than half of the samples for this sequence type. And as you might have heard Nigel French talking about, this is the most common sequence type coming from poultry in recent years in New Zealand. And this sequence type is the fluoroquinolone and tetracycline resistant sequence type. And in a recent source attribution study by Rob Lake and others, including Nigel, they found that ST6964 was the most common isolated from poultry and the 4th most common from clinical samples. And ST50 is also fairly common, and you can see that there are, while 6964 is highly clonal, there are two distinct clades for ST45 and ST50. So now we wanted to make linkages with what is present in the birds with potential vectors and reservoirs. So the first thing we see is that the majority of our bird isolates are 6964 and a third of them are ST50. So we find that many of these isolates from birds, 6964, are indistinguishable from those found from the previous flock, as well as their control flock, which might suggest an on-farm reservoir for this sequence type, for this strain. And there are some linkages between catcher samples as well. ST50 isolates from the current flock, there was linkages between catcher samples as well as the poultry shed, which could have been contaminated from the flock itself. Catcher samples make up the other lineage of ST50 as well as one of the lineages of ST45, and wild rabbits and birds make up the other lineage of ST45. So no evidence that the wild birds and rabbits are contaminating the study flock. So to conclude this section of the talk, we looked into vectors and reservoirs for Campylobacter infection of the flocks and found evidence for carryover of the C. jejuni lineages of 6964 and in one case ST50 from the previous flock. We found no Campylobacter after shed cleanout, however, so there may be an on-farm reservoir somewhere, but it may not be the shed itself. We find high Campylobacter prevalence from the catching crew and equipment. About 44% of those samples tested positive, and the sheds only tested positive after the first cut, after the crew had been in. And there were some isolates that matched closely with the poultry isolates, so these may also be a source of infection. We found no evidence for wildlife, breeder flock, feed, drinking water or litter as a source of infection. And hopefully the newly developed Campylobacter specific metabarcoding approach that Patrick's developing can yield additional and complementary information to that obtained by the cultural isolation and whole genome sequencing. And we expect that reservoirs and vectors for Campylobacter contamination of flocks are likely to vary to some degree by farm location, seasonality and housing system. It might be different for free-range flocks, for example. And ideally, findings from this study should produce information to enable the poultry industry to develop on-farm risk management practices to reduce Campylobacter contamination of flocks. So there are a lot of acknowledgements in bold. We have our study team, and in lighter text we have all the other people that were involved in the sampling and testing of samples. So thanks to everybody involved in this study. And I'm going to move on again to Roy to introduce you to the second study that PNS has commissioned through the centre.
Thank you, Jo. This one is a longitudinal study on the impact of primary and secondary processing on levels of Campylobacter on broiler carcasses and portions. So the three factories selected had processed more than 250,000 chickens per day, about 65 million birds per year, just over half of the total production. So they're significant. So we had a history of completing trials, but fairly fragmented. There was no total industry trial and very few total process trials either. So the project goals there, I don't need to read that out. What we're going to do next is have a quick look at the primary processing and what happens. Now, before we start, just the principles of primary processing, there's two basically. Keep it clean, prevent the contamination of the carcass. Unfortunately, no equipment manufacturer will give more than a 98% performance guarantee. That's not going to work 100%. And if contamination does happen, then wash it off very quickly. A good researcher friend of ours, Norman Stern, told us we had 15 seconds to get it washed off before it adhered, and we would have a lot more problems. So primary processing, scalding is the first significant event after the bird's been slaughtered. They're usually around 2 to 2.5 minutes at 57 degrees to 59 degrees Celsius. This reduces the levels on the carcass by up to 2 logs, from around log 7 to around log 5. Next slide, please, Joe. Pluckers. These are obviously not in use, but if you get that close to them in use, you have real problems. Lots of rotating rubber fingers, removing feathers and the top layer of the epidermis. Unfortunately, the bird is squeezed slightly, and then any fecal matter is expressed and distributed over the carcass. I hope I'm not putting you off chicken, guys. Normally, this causes a slight increase in numbers, and that's why the post-plucker sprays are a critical factor in poultry processing. Next slide, please, Joe. We'll go through these quickly. The vent drill, this drills a hole in the back end of the bird, and then like a hole saw, sort of makes that hole much wider, and then drags that cloaca and the bottom part of the colon out of the bird and drapes it over the back. If this is not operating properly, it can cut the intestines, and obviously when the further processing, the eviscerator spoons go in, they squeeze the pack a bit, and you've got cut intestines, you've got stuff all over the place. Next slide, please, Joe. The evisceration spoons go in and lift the guts out of the bird. As I said before, if we get any damaged guts, that squeezes stuff all over the bird. Again, well-adjusted sprays are extremely important. Next slide, Joe. This shows carcasses on line with the gut packs separated. So the eviscerator machine has separated the gut packs, that's on the top left, and the carcass is just below them. This removes any further contamination. Next slide, Joe. This plant's got parallel spin chills. This is plant D. Most plants have sequential spin chills. It's large tanks of chilled chlorinated water with counterflow of water on birds. This has a significant impact on Campylobacter. 2-3 log reduction is common, mostly around 3. The primary purpose of these is to cool the birds from 40 to 4 degrees C, but they do have a real impact on Campy. Following chilling, the birds are dipped into ASC for about 20 seconds. That's acidified sodium chloride, which on the initial trials was the best way of reducing Campy levels by up to 2 logs, and then transported to secondary processing for cut-up packaging. Next slide, Joe. I think this one's yours.
Okay, over to me. Thanks, Roy. So as Roy said, we were interested in sampling both primary and secondary processing steps from the 3 largest manufacturers, poultry producers in New Zealand. And so they're anonymised, the plants are anonymised as plant A, D, and F. And specifically, we proposed to take samples from 8 different primary processing steps, some of the ones that Roy's talked about, and also 7 products from secondary processing. At each sampling event, we took 5 of each sample type to give a total of 75 samples per sampling event, and we sampled each plant 6 times, so 6 different occasions, to give a total of 1,350 samples. And all of these samples were sent to Marie Callender at the Tegel New Plymouth Lab, and they did an amazing job of enumeration of all of these Campylobacter on all of these samples, these rinse H samples. Marie's lab then sent the isolates, any isolates they attained up to 2 per sample, to Massey University to add in the winter to do speciation for Campylobacter, and a subset of these are going to be sequenced by whole genome sequencing. And the data we get from the carcass rinse H enumeration is also going to be used to produce a quantitative risk model tool by Beverly Horn and Bridget Armstrong here at ESR. And overall, we wanted to establish the numbers of Campylobacter at each stage of processing and determine the impact of processing steps on numbers and types. But before we actually got started with the sampling, we wanted to refine some of the methodologies that we were using. So the current methods for testing the poultry rinsates had a limit of detection of 200 colony forming units per sample. However, a lot of the samples that we expected to get, especially those past the spin chill step, are expected to have lower than 200 CFU per sample. So we developed a method that involved tenfold improvement of this limit of detection, and this was development of this protocol was done by Marie Callender as well as Roy in the Tegel lab. And so what this involved was taking our rinsate, passing it through a coarse filter to remove a lot of the big chunks, and then centrifuging 50 mils of this. You can see at the bottom of the centrifuge tube the pellet that ideally contains all the Campylobacter in the sample, as well as residue from the carcass itself. We then decanted off 45 mils of this and resuspended the pellet in 5 mils and plated this to our enumeration media. And Marie's lab compared this method against the standard NMD method and could detect Campylobacter from 2.8 more samples on the samples that she tested. So, we did quite a bit of testing of this new method. And depending on the sample type that we were testing, we had 3 different sample testing regimes. For the samples that are expected to have high numbers of Campylobacter, the carcass was placed into 400 mils of buffered peptone water, and it was plugged to stop the cecal and fecal contents coming out into the sample. And we did a 2-minute rinse on those. And then the limit of detection from plating was 400 CFU per carcass, and we plated quite a lot of dilutions for those, or I should say the Tegel New Plymouth lab did. For the whole birds post-spin chill, again, we had our 2-minute rinse with of the carcass with the buffered peptone water, but we applied a new centrifugation method to concentrate up the Campylobacter in the sample for a limit of detection of 20 CFU per carcass. And portions were sampled slightly differently. We had 1.8 kilograms of portions per sample added to the 400 ml buffered peptone water. And the rinse in this case also involved a massage of the portions together for 2 minutes. And again, we used a new concentration method to get a limit of detection of 20 CFU per sample for these sample types. And we trialled all these proposed methodologies first in a pilot survey, which involved 1 sample per sample type at each of the plants. So this also not only refined our methodology, but it also served to train the samplers at each plant as well. So Roy was heavily involved in that. So the questions we wanted to ask during primary processing were, what is the impact of each primary processing step on Campylobacter numbers? So the overall processing steps are the same between the plants, but there are various differences. For example, the use of equipment from different manufacturers. So we wanted to know, are there also plant-to-plant differences at each step? And we wanted to know how this compares with a 2013 study, which only looked at primary processing and was carried out on plant D. So this is the data from primary processing. This is an initial look at the data displayed as a violin plot. So if you're not familiar with violin plots, the blue dot, I'm sorry that my pointer's not working, but the blue dot in the middle is the median observation for that sample type in that plant. And the width of the violin is, the wider the violin is, the more observations of Campylobacter at that concentration there is for that sample type. And the red dotted line is the limit of detection for each sample type. And you can see the processing line, and we're starting from the left, going to the right for the processing. So the first thing that we see is that there's at least 2 log reduction between pre-scald and the post-scald step. And we also see at least a 3.5 log reduction going between the post-inside-outside body wash to the post-spin chill step. And many of these post-spin chill and post-acidified sodium chloride steps are below the limit of detection, which was 20 CFU per sample. And strikingly, 76% of our post-ASC samples had no detection, so less than 20 CFU per sample. And 97% of these had less than 200 CFU per sample. So this was the limit of detection of the previous NMD method, or the standard NMD method. So this also shows the benefit that our new centrifugation method brought to the study. And we're also exploring, one of the next steps is to explore plant-to-plant differences. So you can see, for example, in the post-SCOLD step here, there's about a 1 log difference between plant D and plant F for the median counts. And there's also differences in distribution of the counts as well. So the data in plant A look like they're more compacted, so it could be that this is more consistent in plant A, but that's something that's going to be explored further. We also compared the data from plant D with that obtained in 2013. So the other plants that we sampled in this study weren't sampled in 2013. And the methodology was mostly the same, except the limit of detection was different at the post-spin chill and post-ASC steps. So you can see that for most steps, actually all along, the Campylobacter mean log counts were lower from the 2021 study compared with the 2013 study. And because the limit of detection is a little bit different at post-spin chill and post-ASC for the 2 different years, it's a little bit, looking at the graph is a little bit arbitrary, but what we do see is that 18% of the post-ASC samples in 2013 had no detected. So in other words, 18% had less than CFU per sample. But we find in 2021, 100% of the samples have less than 400 CFU per sample. And in fact, 77% of the post-ASC samples in 2021 had less than 20 CFU per sample. So that really suggests quite a significant improvement between since 2013 for this plant, and it's likely that similar changes are occurring across all plants as well. So moving on to secondary processing. The secondary processing such as portioning, deboning, and deskinning can also influence Campylobacter prevalence in numbers on the product. And this could occur by cross-contamination or perhaps reduction of hospitable niches. So if you're removing the skin and the feather follicles where the Campylobacter is hanging out during deskinning, how's that affecting the Campylobacter levels on the portions? And there's also evidence that Campylobacter can survive less well on skinless poultry than on skin surfaces. So we were interested in, does Campylobacter levels differ by product type, and are there plant-to-plant differences? So I'm showing you now a violin plot of the samples from secondary processing. And the first thing that we see is plant F on the bottom has higher counts on particularly on whole bird samples than the other plants. And indeed, plant F whole bird numbers are much higher than what they had at the end of primary processing. And so the difference between the plants here, we think, is because plant A and plant D have secondary and primary processing occurring at the same site in the same plant. So the carcasses, whole birds are going pretty much directly from primary to secondary processing. Whereas plant F, the whole birds are going into a bin and the secondary processing is in a different location. So I think there's several hours between primary and secondary processing for plant F. So there's a higher chance for cross-contamination of those whole birds. The next thing that we see is that Campylobacter numbers are higher on most of the portions, except for the drums compared with the whole birds. And some of the reasons behind this might be that due to different sampling methods for the portions compared with the whole birds. So if you remember, I was saying that for the portions, the rinse involves a massage, so that could involve friction between the different portion types, and that may be washing more Campylobacter off. It could be also breaking up clumps of Campylobacter that's present. In addition, the portions come from more than one bird. So for example, the wings, there are 17 to 18 wings per sample, so that could be up to 17 to 18 birds that these are coming from. So there is a higher chance of cross-contamination if there's higher chance of them arising from a bird that had higher numbers of Campylobacter than you get for a single carcass. But the thing is, these are also true for the drums as well. So we're not sure why drums have lower Campy counts than the other portions. And some of the portions also have higher surface area to volume ratio. So that's another comparison that we're going to be looking at as well. Some of them have a lot more skin, for example, wings. And another thing that I found interesting, or we found interesting, is that we expect the highest numbers of Campylobacter on the skin, and because it's present also in the feather follicles. However, deskinning the products did not necessarily decrease Campylobacter loads on the product. So you can see in plant A, compare breast skin on with breast skin off, and there was actually higher counts on the skin off products than there was from the skin on products. So that might suggest some cross-contamination is going on during the processing. And we're going to make additional comparisons, for example, taking surface area into account and the number of skin per sample into account, as well as looking at Campylobacter numbers per serving portion. So to give a summary of our results so far, this is still ongoing because the sampling was only carried out earlier this year. We developed a rinse rate concentration method, which had 10-fold improved limit of detection. And this is really beneficial for our survey because 24% of the samples had between 20 and 200 CFU per sample, and 200 CFU was the limit of detection for the standard NMD method. The current primary processing steps resulted in 6-fold reduction in Campylobacter numbers, and there was no detection in in other words, less than 20 CFU from 76% of our samples at the end of primary processing. So this is that's a pretty significant reduction. And we see measurable improvement in Campylobacter reduction during primary processing compared with, for plant D, compared with what we saw back in 2013. For secondary processing, we're seeing plant-to-plant differences for Campylobacter numbers on product, and we see lower numbers of Campylobacter on drums and other portions, and higher Campylobacter numbers on other types of portions compared with whole birds. And so the data is going to be used to provide a quantitative risk model to assess the impact of improvements of individual steps on Campylobacter numbers. This is being done by Beverly Horn and Bridget Armstrong here at ESR. And Patrick and Anne has done speciation of a lot of the isolates, and Patrick's going to be carrying out whole genome sequencing on a subset of the isolates to assess the effect of antimicrobial processing steps on the types of Campylobacter present on the product. And the data will also be able to provide a baseline from which to gauge the efficacy of future intervention strategies. So I'd like to acknowledge all the people involved in this work as well. Again, involved project team and Enlightened Text, all the people involved in the sampling as well as the processing of the samples. And Roy, did you have anything to say, or else we'll just move on to questions?
Just very quickly, over 2.5 tons of chicken was rinsed or massaged during this project. And the laboratory organisation for the logistics of sampling and testing of the 1350 samples was superb. Excellent, well done, folks.
Great, thank you both, Joe and Roy. That's fantastic. So if anyone's got some questions, please either throw them onto chat or just indicate that you'd like to speak. I do have a first question. One of the questions I have is that, I think I learned an amazing stat that half of the poultry processed in New Zealand is whole birds that's processed or cut up at the supermarkets. So the finding that your whole birds have higher levels, I was just wondering, have we got plans to move into doing some more work at the supermarket level as well? Or what are the implications of that finding?
I think the Campylobacter group within FSSRC have been discussing that, and I think that will happen hopefully in the relatively near future.
What do you think, Joe? You think it's quite a significant.
Yeah, I think it is quite interesting, especially because some of the other processing, when the whole birds are going off to be processed by like supermarket chains, for example, they're also going in the big bins, the same as what happened for plant F. So that might be, we think that is an interesting area to look at as well. And that is something that, as Roy said, is being discussed.
Okay, great. I've got a question from Dean, who's asked, could ASC be used on the portions to reduce Campy numbers?
Yes, it can, and it was used extensively in the USA for a few years. The issue is that the portions come out in many different areas of a plant, so you need many different ASC treatment baths. Whereas whole birds, they all go through one. So it is possible, and I think we as an industry probably ought to trial it. And I know Michael's been talking about it for a while. So we'll have a look. It'll be interesting to see how much impact that does have.
Okay, yeah. Any other questions? Patrick, did you want to comment on some of the work you've been doing on the metabarcoding, which sounds pretty exciting? Patrick might not be there anymore.
No, I'm still here. Hi, yes. Yes, but no. I'd kind of leave it because I'm still working on stuff at the moment, and there are some interesting results coming out, and I'll just leave it as a tantalizing thing like that for now.
Okay, great. No, it's fantastic. It's great work that's been done. Okay, there's a question from Gail. We've seen that if reduction in flocks could be achieved, particularly by improving catcher and equipment, you could lower the risk significantly and may not even need the ASC intervention at the end of processing.
No, unfortunately not. The issue would come as we saw much of the introduction of Campy to the flocks was from elements that had been on the farm from the previous flock. When I say on the farm, because it may not have been in the shed, it may have been in the farm environment somewhere and got carried into the shed. So catching on its own will have a marginal result. It's understanding that whole thing around Campy on farm that's going to have the major one, I think.
Yeah, I agree. Yeah, because there's going to be multiple vectors and reservoirs as well, and it may be different farm to farm as well.
Yeah, I guess the variability in the farms and what's happening in some of the management is also an area. But there's a question from Kath. Thank you. She says superb science. Interested in whether you have future plans around the findings from this farm study. Are there some practical options to reduce the potential cross-contamination coming from the catchers and equipment, or are there plans to investigate other sources on farm?
The industry itself has two proposals in front of them at the moment,
okay,
which we need to refine before we come and talk to the wider team.
Yeah. Yeah, and Gail makes a comment, ideally if flocks could be cleaned up, your risk would be have a big reduction.
Yeah, no, Gail's right. You know, we did this with salmonella as I started.
It made a huge difference.
But all of the things we've tried on farm, and there've been an awful lot of them, and MPI have tried some as well, such as caprylic acid, have had very, very minimal impact.
Okay.
Much harder to control on farm than salmonella was.
Gail's just made, no, not Gail, David Lowry's asked, given that drinking water has previously been shown to be a significant vector for salmonella, do you have any thoughts on why this is not the case for Campylobacter?
Okay, it's not the case for salmonella anymore either. The drinking water is chlorinated at source, and then when it arrives at the farm, they use chlorine dioxide to further treat it. In fact, no Campylobacter in the drinking water or in the water anywhere on the farm.
Yeah. We're going to have to wrap up, I think. Someone's just asked whether we get a copy of the PowerPoint presentation. We do host these the recording, and it's put on our website, and Michael sends out links. So just actually, I'd ask you all to, before I thank Joe and Roy again, just ask you to fill in the survey that comes out after this. We're just trying to collect some ideas on other seminar topics and make sure that we're meeting some of the needs of interested parties. So, so thank you very much, Joanne and Roy, for a really great talk. I think it's been really interesting, and it's fantastic. The study is drawing in so many people around such an important topic. Really appreciate your work. It's really good. And just to let you know, the next seminar will be the last week of April. We're sort of because of Easter, we've run it a bit short. So we're on the 30th, the last day of April, which is 30th of April, and we're hoping to have a topic around the economics of food safety recalls. I'm just trying to nail down the speaker on that. So looking at the impact of that on industry. So thank you once again, everyone, for calling in. And oh, one more question, I think. Someone just says, thanks for the great work and presentation. May I ask if there's some record in terms of the odor of ASC and similar sanitizing agents from consumers in New Zealand, and some early conversations about changing the agents in industry? This is from Stanley at CSIRO in Australia.
Okay, ASC has no odour. The concentrations we use it on, you cannot smell it on the bird even when it freshly comes out of the tank.
Yeah.
And it depletes very quickly. You know, within an hour you can't detect it on the surface of the bird.
Yeah, so I also had a question about the change of methodologies. Do you have to go through a process of getting that sort of confirmed as an acceptable? So that's still got to be done?
No, ASC, certified sodium chloride, is the Fazan's approved processing aid.
Oh, no, I mean the methodologies that Joe talked about for limited detections. Is that true, Joe?
Right. Yeah, so we are undergoing higher level validation with that now with MPI, so that is ongoing. Yeah.
Great. Okay, well, thank you, everyone. This has really gone the full time, and it looks like everyone's very, engaged, and we've had lots of questions. So thank you very much, and I'll send out an email, and just remember to fill in that survey. Thank you.
Thank you.